Hello, I'm Neil Clark, and this is the fourth and final part of a series of talks on the characteristic direction, which is a new geometrical approach to the analysis of differential gene expression. In the previous part, we looked at some validation of this approach and comparisons to other approaches in the context of transcription factor perturbations and drug perturbations, using microarray data. Here we wanted to address the question of whether this also might be a good way of looking at gene expression in the context of new high-throughput sequencing technologies. In this case, there are fewer data sets to work with, but we've found we've tested one which derives from this paper studying the role of STAT3 in the mechanism of tumorigenesis in large B-cell lymphoma. And we're going to compare the characteristic direction approach to the most popular approach in the context of high-throughput sequencing, which is called a method called DE-seq. So here we've taken data from this study, which is examining differential gene expression between two subtypes of large B-cell lymphoma, and also differential binding of this transcription factor STAT3. And one of the things that they did in this paper is they looked for consistency between differential binding of STAT3 and differential gene expression. And what we did here is we reanalyzed the data using the characteristic direction and compared to the analysis with DE-seq. And so what we have here on the right is a plot of a similar kind to the one in the previous talk, which sort of quantifies the degree to which genes which are differentially bound by STAT3 are also differentially expressed, broadly speaking. And you can see that the characteristic direction prioritizes those genes differentially associated with differential DNA binding of STAT3 to a much greater extent than DE-seq. So this further sort of provides confidence that this is a good method to use, even in the context of high-throughput sequencing. Finally, we're going to look at compare the performance of these methods with synthetic data. As we said earlier in the previous talk, this is not applying this to synthetic data has the problem that the synthetic data only has the structure that we give it, so it's not as meaningful a comparison as what we've shown previously. But for completeness, we include it. And so we're looking at the prioritization of truly differentially expressed genes. We know they're truly differentially expressed because we told them to be. And we see even in this context, the characteristic direction is outperforming the other methods in the recovery of the differentially expressed genes. So you can read these. These are receiver-operated characteristic curves. So, in summary, we've described a new approach to differential gene expression called the characteristic direction, and we've shown that it takes into account the cursive dimensionality. It tries to sort of regularize and address that problem, but it tries to use the structure and the data in a multivariate way to characterize the differential expression. And also it provides, one of the reasons I like it provides a nice geometrical conceptualization of differential expression. You can imagine the difference between two states is sort of characterized by a direction, the direction between them in expression space. And finally, this geometrical approach has some very natural extensions, which we're going to talk about in the next talk. It's an approach to differential gene set enrichment analysis.